Hi, this is C Sumathi, Assistant Professor in PG and Research Department of Biotechnology, JJ College of Arts and Science, Pudukkottai. Now here going to discuss about glycolysis. Glycolysis. Glyco means carbohydrate or sugar. Lysis means breakdown. So this is the major pathway, major metabolic pathway. In 1940, glycolysis became the first major metabolic pathway. Biochemically, glycolytic pathway means oxidation of glucose to pyruvate with the generation of ATP and NADH molecules. And one more point, it is an universal pathway because it is present in all organisms, in eukaryotes and also in prokaryotes. In eukaryotes, glycolysis takes place in the cytosol. The glycolysis derived from the Greek word glyx, sweet, lysis means splitting. I already said. What is the history of glycolysis? It is the most common pathway which was discovered by the scientists Gustav Embeden, Otto Meyerhoff, and Jacob Carol Parnas. These are the scientists discovered the glycolytic pathway. The biochemical experiments in glycolysis started with the experiments of French scientist Louis Pasteur in the year of 1850. Louis Pasteur in the year of 1850. The discovery of the pathway took almost 100 years of fully unravel. The mechanism was originally succeeded from the curiosity to understand the fermentation process so that only the better wines could be made. Then next, the definition of glycolysis. Next is the definition of glycolysis. Glycolysis is a metabolic pathway where a 6 carbon molecule of glucose is broken down into a 3 carbon molecule called a pyruvate in a series of reactions, in a series of biochemical reactions, that is especially oxidizing biochemical reactions. The byproduct of these reactions is the release of free energy, that is, which is stored in the high energy phosphate bonds of ATP and other reducing equivalents such as NADH or NADH plus that are further used to produce a more ATP in the mitochondria. A glycolysis takes place in both in the presence of oxygen and in the absence of oxygen. In the presence of oxygen, it is aerobic glycolysis. In the absence of oxygen, it is an anaerobic glycolysis. The lactate is the end product of anaerobic glycolysis. In aerobic condition, pyruvate is the end product. Further, it is again converted into carbon dioxide and water molecule. The high energy phosphate molecule of ATP is released during the glycolysis reactions. It is an energy yielding pathway. Glycolysis pathway is also called as an energy yielding pathway. The net reaction of the glycolysis is glucose plus 2 NAD plus 2 ADP molecules plus 2 phosphates, inorganic phosphate. It gives 2 pyruvates plus 2 NADH molecules plus 2 ATP molecules. This is the net reaction of glycolysis. There are a lot of reactions occur in the glycolytic pathway. It is occurred in a sequential manner. Totally there are 9 reactions takes place in the glycolysis pathway. The glycolysis pathway differentiated into two phases. Occurred in two phases. The pathway occurred in two phases. One is preparatory phase, and the next one is payoff phase. In the preparatory phase are again divided into two stages. First stage is stage of phosphorylation, then second stage is stage of splitting. The preparatory phase means in the glucose molecules prepared for the entering into a glycolysis pathway. In the first stage of phosphorylation, the net energy is spent, that is ATP is gained in this pathway, gained in or utilized in this stage. ATP molecule is utilized in this stage. Then stage of splitting, the second stage of splitting, the 6 carbon compound of fructose is splitted into a 2 3 carbon compounds. This is the stage of splitting. The next phase, second phase, payoff phase. Payoff phase means the generation. In the first phase, the net energy is utilized. The second phase, the net energy is generated. Here, the ATP molecules and NADH and NADH plus molecules are generated. In this slide, we will see the total pathway of the glycolysis. The 9 reactions where it is ATP is utilized, which enzyme is catalyzed that particular reaction. This is the whole pathway of the glycolysis. Totally 9 reactions is there. In the coming slides, we have discussed each and every step of the glycolysis pathway. The first reaction is occurred in the preparatory phase, the first stage of phosphorylation. The first reaction, glucose is converted into a glucose 6-phosphate. The glucose molecules is converted into a glucose 6-phosphate. This reaction is catalyzed by the enzyme glucokinase or exokinase. Glucokinase or exokinase. This is the energy requiring step. So one ATP molecule is consumed to phosphorylate the glucose molecules. To phosphorylate the glucose molecules. The second reaction, the glucose 6-phosphate molecule is further converted into another hexose molecule of fructose 6-phosphate. Glucose 6-phosphate is converted into a fructose 6-phosphate. This reaction is catalyzed by the enzyme phosphohexoisomerase because glucose is the isomer of fructose molecule. So isomerase enzyme is catalyzed the above reaction. The next reaction, the reaction 3, the fructose 6-phosphate is converted into fructose 1,6-bisphosphate. Once again, the sub-product is phosphorylated. The enzyme phosphofructokinase is catalyzed the above reaction. Here also one ATP molecule is utilized to donating a phosphate molecule. To donate the phosphate molecule, this is a rate limiting step in the glycolysis pathway. The enzyme is also used in the regulatory mechanism. The next reaction is fourth reaction. First 3 reaction comes under the stage of phosphorylation. Then here it is a stage of splitting. That means 6 carbon compound is split into a 3 carbon compound. Glyceraldehyde 3-phosphate, 1 aldehyde compound and 1 ketone compound, dihydroxyacetone phosphate. This is a ketone compound. Here the 6 carbon molecule is splitted. So this phase is called as a splitting phase. Fructose 1,6-bisphosphate is a 6 carbon compound which is in this step splits into a 3 carbon compounds by the enzyme of phosphotriose aldolase. Phosphotriose isomerase, which is the enzyme which the glyceraldehyde 3-phosphate and dihydroxyacetone phosphate both are isomerase. Both are isomeric compounds So this reaction is catalyzed phosphotriose isomerase. Phosphotriose isomerase. Then next, second phase, payoff phase. First phase is preparatory phase, second phase is payoff phase. In this phase of glycolysis, energy is yielded. In the first phase, the energy is utilized. ATP molecule is consumed, utilized. But in this phase, the energy is yielded, energy is generated. In the fifth reaction, glyceraldehyde 3-phosphate is converted into a 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. During this step, one inorganic phosphate is utilized and one NAD plus get reduced to form a NADH plus H plus. Then again, 1,3-bisphosphoglycerate is converted into a 3-phosphoglycerate. Here the enzyme 1,3-bisphosphoglycerate or glycerokinase is involved to catalyze the above reaction. This is the enzyme, the only reversible kinase of the entire glycolytic pathway. Here ADP is converted into a ATP molecule. Here ADP is converted into a ATP molecule. This is in this step one ATP molecule is generated. Then 3-phosphoglycerate to 2-phosphoglycerate. Here the phosphate group is mutated where the third carbon compound having the phosphate in the 3-phosphoglycerate molecules. But in the product 2-phosphoglycerate, the phosphate molecule which is present in the second carbon. So we are naming 2-phosphoglycerate. So the phosphate group is mutated in the product. So the enzyme phosphoglycerate mutase is involved in the reaction to catalyzed. The next eighth reaction, the 2-phosphoglycerate is converted into a phosphoenol pyruvate. It is simply called as, it is also called as PEP. It is also called as PEP by the enzyme enolase. During this step, a water molecule is released. At last, we comes to the last step or the end of the glycolysis pathway. The phosphoenol pyruvate is converted into a pyruvate molecule. Phosphoenol pyruvate is converted into a pyruvate. It is a high energy compound. It is a compound which is next get into another pathway. The pyruvate molecule is a starting point of the TCA cycle or otherwise called as Krebs cycle. This opposite reaction is catalyzed by pyruvate kinase. Mostly the enzyme kinase involved that denotes the involvement of ATP molecule in the reactions. Here the ATP molecule is yielded. Here the ATP molecule is formed. One ADP molecule is converted into a ATP molecule. Then the energetics, then we discuss about the energetics of glycolysis. Energetic means how much of energy generated after the glycolysis pathway completed. The energy produced by glycolysis is different under aerobic condition and anaerobic condition. So because under aerobic conditions there is an extra generation of 2 NADH molecule that are further oxidized inside the mitochondria to produce ATP. Generally the oxidation of NADH within the mitochondria is responsible for the generation of 2 and of ATP molecules per NADH molecules. However, under anaerobic condition there is no net generation of NADH. In this image, just we are having an overall assumption of energy yielding. First, glucose to fructose 1,6 bisphosphate, 2 ATP molecule is involved. 2 ATP molecule is involved. Then again in the payoff phase, 2 ADP molecule is involved, 2 ATP molecule is generated. Like that, 2 NADPH molecule is 2 NADH molecule is generated. 1 NADH molecule is equal to 3 ATP molecules. It's approximately equal to 3 ATP molecules. The fructose 6 phosphate, fructose 1,6 bisphosphate is a 6 carbon compound. In the stage of splitting, 2 3 carbon compound is formed. Every 3 carbon compound forming a 1 pyruvate molecule. So total are 2 pyruvate molecule we gained. So again and again there is a two sequence of reactions is take place. So totally uh, we getting two NADH molecules and two ATP molecules. So more or less 8 molecules of ATP molecules gathered after completion of a glycolysis pathway. After completion of a glycolysis pathway. These are the overall energy gained up to the level of glycolysis pathway. There are three regulatory enzymes which is involved in the glycolysis pathway. One is hexokinase, second one is phosphofructokinase, third one is pyruvate kinase. Hexokinase is the enzyme catalyze the reaction glucose to glucose 6 phosphate. This enzyme is inhibited by glucose 6 phosphate molecules. The phosphofructokinase is inhibited by ATP, citrate and H plus ion in the low pH value. Then which is activated by fructose 2,6 bisphosphate, AMP, fructose 6 phosphate molecules. At last, the pyruvate kinase enzyme is inhibited by ATP and acetyl-CoA and it is activated by fructose 1,6 bisphosphate and AMP. Adenine monophosphate. By the above glycolysis pathway slides, we just get an overall idea about the glycolysis, how is the reactions takes place, what are the stages present in the glycolysis pathway, then how much of energy we gained through the glycolysis pathway, and what are the regulatory enzymes in the glycolysis. These are all the overall view of the slides. That's all about glycolysis. Thanks for listening. Thank you. Thank you so much.